Welcome everyone, and thank you for joining us for today's webinar, Digital Biosensors and the Internet of Biology, presented by Dr. Brett Goldsmith, CTO of Cardea. My name is Susie Valdez, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational events and webinars. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have any trouble seeing or hearing this presentation, simply click on the Ask a Question box and let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process to obtain those credits. Without further ado, please join me in welcoming our presenter today, Dr. Brett Goldsmith. Welcome, Dr. Goldsmith.
Thank you. Without further ado, I will get started on the presentation. It's my pleasure to talk to everyone. I'm going to be talking today about digital biosensors and the Internet of Biology. And there's some words in there that are probably unfamiliar to you, and that's okay. My name is Brett Goldsmith. I'm the CTO and co-founder of Cardea, and I am a nanotechnologist and a physicist. So that may color some of the talk that you're about to see. To begin talking about the Internet of Biology, I want to start with biological networks. Biological networks are something that we're just now starting to understand. They're the combinations of single-celled organisms all the way up to people that communicate with each other through chemistry and biochemistry, passing messages and energy back and forth. These are cell signaling networks and things like your immune system and your microbiome. On the left here, we have a rough map of the human microbiome and then a zoom in of just the gut microbiome network. And these are really very complex things. Biology is very complex. The inherent properties of these networks are that they're very local, hyperlocal. They communicate via chemistry, so you have to wait for the chemicals to go from one place to another. They're also dynamic. They change from one moment to another. If you wanted to get a snapshot of something, it would be pretty hard to see the same shape and elements of the network from 1 minute to the next. And they're self-organized. This is not something that's engineered. It's something that comes naturally out of the interactions between all of these biological components. Despite that self-organization, you can see some structure in here. They're not random collections of things interacting with each other without any sort of order. There is some order here. But probably the most important thing about these networks is that you do not have access to them. If you wanted to know right now whether or not your microbiome is detecting any pollutants, or if your immune system is fighting off the beginning stages of a cold, you don't know. The best you can do is you can get a snapshot by sending away some samples to a lab, and you can find out in a couple of weeks. But this is a series of networks that we as humans don't really have access to. In contrast, digital networks are how we all share information. We're all familiar with the internet. It's, again, contrasting with the local biological networks, this is a global network. It spans the earth and beyond. It's also instant. So you get to know information right away. If you want to look up the population of Denver, you can find out the population of Denver in a matter of moments. It's also engineered, and it's human readable. Very, very important that things be human readable. Otherwise, we don't get to interact with them. Despite the big differences, the structures of these networks, biological networks and digital networks, are similar. They're both exponential scaled networks with some order within them. But to truly utilize biological networks, we need to find a way to to get them access to our digital networks. Again, this is how we share information. If something doesn't have access to our digital network, if your immune system doesn't have a way of getting information into the internet, then you're stuck in an old 20th century approach towards understanding biology. This idea of coupling digital networks with biological networks is the Internet of Biology. And this is something that's talked about out there by several different people. And it's that idea that you're integrating the biological networks that are local with the internet, which is global. You're taking something which is dynamic and also providing instant access to it. And you're able to take a very complex engineered network and link it to a complex emergent network. So you might want to use the Internet of Biology to ask questions like, am I getting sick right now? Is this food right here healthy? Is this environment clean? And the information that's flowing from organisms, cells, and people that interact chemically and biochemically with the environment should be able to tell you the answers to those questions right away. Now, doing this connection, actually connecting the chemical, relatively slow biological networks with the instant global network of the internet is very difficult, and it requires some gateway. That's where Cardea comes in. We don't actually make the Internet of Biology. We build the network gateways. And so that's going to be the primary thing I'm talking to you today about. The biochemists that we work with build the apps on the Internet of Biology, and the engineers that we work with build the tools that allow you to interact with the Internet of Biology. But we build this gateway in between. To describe a little bit of elements of these different worlds that interact together, we can look through some of these areas here. So for example, wireless networking allows that global communication and the local portability of the internet. The combination of electronics fabrication and personal devices allows you low-cost access to the internet and low-cost access to the information wherever you are. Machine learning provides very powerful tools for you to understand information that might take you a long time to study otherwise, providing a little bit of extra computational oomph to your brain. On the biological side, genomics provides information about what biology could be or might be. Proteomics provides information about what is currently going on in biology. The microbiome, as an example, is a local biological network that is an information exchange and exchanges all sorts of different things with agricultural networks and your own wellness. Synthetic biology is how we engineer biological tools in this area. I'm a nanotechnologist. I don't build nanobots. I do something much more powerful. I work with synthetic biologists. So if we're working on the gateway in between the biological networks and the digital networks, what is that gateway? That is a digital biosensor. So what is a biosensor? There's an easy definition for a biosensor. It's simply a device that connects biological or converts a biological interaction into a machine-readable signal. Now, typically, this is done without the use of extra reagents like a label, generally. In this kind of picture, which I like, this is a version of a picture that's been floating around in the nanotechnology community for a long time. The biosensor is this alligator clip. And if you can think of it like there's a wire that stretches out from your computer or your smartphone and just clips onto a protein and tracks what it's doing, the biosensor is that alligator clip. It's the thing that allows you to see what the protein is interacting with in the environment. Is it active? Is it not active? Is it binding to something? Is it not binding to something? So understanding really how a biosensor works, we want to start with the anatomy of a biosensor. This is important and often misunderstood. Biosensors are made up of multiple different pieces. There's the receptor, the transducer, and the computer, which also includes all the software and the way that we look at the data and understand it. The receptors here at the top, I've drawn my physics versions of proteins, which are those little orange Pac-Mans. Now, the receptor is the component that actually interacts with the environment. In a modern biosensor, this is something like an antibody or DNA, but in the beginning of biosensors, this was most often a polymer. And in reality today, it's usually a combination of synthetic and biological components. This is the part of the biosensor that gives it its specificity and sensitivity. It tells you how well your sensor is able to detect the one thing you're looking at without getting distracted with a false positive, false negative, and it also tells you how low and how high concentrations you can measure. The key for the rest of the biosensor system is to work well with this receptor and to get the most out of the chemistry that it brings. But it's not really realistic to think that your device is going to improve upon your biochemistry. So it's very important that this receptor layer be made well and be specific for the application you're trying to develop your biosensor for. The transducer is the technology component, and often when you talk to engineers, they'll talk about this as being the biosensor, but in reality, it's just part of the biosensor. And there's a lot of different kinds of this, electrochemical and some of the other ones I've listed here. So some of the things that you really want to think about when you look at the different technologies of transducer is what kinds of receptors can be used on this transducer, because not every technology is compatible with every type of biology. How is a receptor coupled to the transducer? Is this something that can be done easily, or does it require a lot of work? Is this the kind of thing that's going to be good for one type of protein or one type of molecule, or can it be used generally? And do you get the full range of sensitivity and selectivity when you're using a particular transducer with a particular set of molecules? Does the technology itself limit your reading in some way? These are the kind of things that people think about when they're designing a biosensor. And in the past, these are the kind of things that have limited the use of the biosensor. In particular, the different technologies really influence, or rather control, the size and the cost and the overall capabilities of the system. So if you want something that's portable, a lot of technologies aren't going to be appropriate. If you want something that's inexpensive, a lot of technologies aren't going to be appropriate. And so you really need to figure out exactly where do the different technologies fit. The last part is the computer, and this is what provides access to the rest of the world, to your biosensor. And theoretically, every biosensor today can make use of powerful computational tools, but the reality is that that's not how everything works. And some biosensor technologies fit better with computational tools than others. One more little bit of history here. There's just a little bit of history on biosensors. This is actually old technology. It started in 1956 with the Clark electrode. This is something that allowed doctors to measure oxygen levels in blood in real time, and that revolutionized surgery. So you can see here, most of the commercialization in biosensors was done relatively recently. So 1990 was when SPR, surface plasmon resonance biosensors, were commercialized by Pharmacia Biocor. This was the first big use of biosensors outside of the electrochemical biosensors that were first developed in the 50s. And since then, there have been commercial launches of fiber optic biosensors and now field effect biosensing biosensors. It's taken this evolution of different biosensors to get to where we can talk about the Internet of Biology and digital biosensors. I'm going to describe a little bit of why it is that we need this new FEB type sensor to talk about these things. To start off with, electrical biosensors, the electrochemical biosensors that were first developed, are by far the most common type of biosensor out there. So typically, these biosensors, like the Clark electrode, they measure things like salt or dissolved gases. But the major exception here is probably the most common biosensor in the world, and that is glucose monitoring. So most of the monitors that you use to check your glucose level if you're diabetic rely on some sort of electrochemical biosensor. And so they're actually all over the place. They're inexpensive, relatively easy to use. Obviously, they can be handheld, relatively fast. The major drawback of an electrochemical biosensor is that it requires some sort of charge transfer. Basically, you need an element of biology that acts like a battery. And that can work when you're trying to measure oxygen or glucose or salt, but it doesn't work with most biology that's interesting or useful. It also generally requires relatively high concentrations. Now, even with all of those limitations, you can still see very powerful biosensor systems like the iStat, which is a handheld biosensor from Abbott, I believe, that allows you to do a lot of different handheld biosensing measurements. It's not quite exactly what we're talking about for a digital biosensor, though, because you're not getting a lot of complex biological interactions. You're getting a snapshot of some of the more energy transfer related interactions of biology. A more general biosensor would be, in the past, something that would be an optical biosensor. And what I'm showing here is a diagram of an SPR system, surface plasmon resonance. This is a really big system, really complex to use, really expensive, and that's typical of a lot of optical biosensor systems. Optical biosensors are very powerful. They allow you to measure almost any biochemical interaction. When they were first launched, they were generally only capable of measuring large protein-protein interactions, but a skilled user can measure just about anything these days. They're very popular in research labs and early stage drug discovery in particular, but that's because that's where you can find the people with the expertise and the budgets to use them. They're not the kind of thing that you're going to carry around in your pocket. They're much, much larger. Digital biosensors combine the benefits of the old style electrochemical sensors with the capabilities of optical biosensors, meaning a digital biosensor is relatively low cost, relatively fast and easy to use. It can be handheld, but it can also measure any biological interaction. So now you have a tool that allows you to look at protein-protein interactions, small molecule interactions, the dissolved gas and salt interactions, basically anything that a cell might be interested in can be monitored by a digital biosensor, and it's in a package that you can carry around in your pocket. So we now have access to portability and a large biological range of measurements. This was only possible because of nanotechnology. And this may be actually the first real enabling technology created by the nanomaterial boom that started at the start of the 20th century. I am a nanotechnologist, so at this point I am honor-bound to give you a brief history of nanotech in this area. Around 1990 was when field effect biosensing, the technology that drives digital biosensors, was first demonstrated in a pre-nanotechnology world. It was first used with silicon transistors. And while people were able to show that silicon transistors could be used to measure biological interactions, they also were able to show pretty clearly that silicon was not going to work from a practical point of view in any sort of device. And the reason for that is silicon just does not play well with salt water. And salt water is unfortunately the environment that most biology likes to live in. The corrosion that happens to the silicon material when it's put into blood or any biological fluid creates a barrier between the silicon and the interesting biology that you want to measure. The result is that people were frustrated and basically decided this technology, field effect biosensing, was stuck until a new semiconducting material could be found that would be able to operate in salt water. About 10 years after that, the first nanotransistors were made, and one of the first applications of these transistors was biosensors. And these used carbon nanotubes. Carbon nanotubes, unlike silicon, are stable in salt water. you can take a carbon nanotube transistor and put it in whole blood, and the transistor will still function. This was a really exciting time to be a nanotechnologist. We were developing all sorts of really interesting measurements, and we were seeing things in protein structure, biochemistry, and real-time dynamics of biosystems that no one had ever seen before. We got to publish papers in great journals. We got to go to conferences and give talks to packed rooms. And one of the reasons why we were able to do that is no one else could use these devices. The problem with the first generation of nanoelectronic digital biosensors is that they were only usable in a nanotechnology lab. Carbon nanotube transistors were very difficult to make, and the technology couldn't be transitioned to a biolab. Only the biologists that happened to be able to collaborate with a nanotechnologist could make progress. So that led to some disappointment and a loss of interest. Until about 10 years later, graphene biosensors were demonstrated, and this led to the second explosion of nanoelectronic biosensors. There was, again, a lot of excitement over what could be measured. We did, again, a lot of really great measurements. But this time we also felt like there was potential to reach beyond the nanotechnology labs because we believed graphene could be manufactured and turned into something that would operate outside of a nanotechnology group. Something manufacturable and commercializable. And in 2016, we got to the first use of a commercial nanosensor, a real digital biosensor, in a biology lab. I want to tell you a little bit about that experience because I think this is the first time that a digital biosensor was actually used by a biologist. So in 2016, we had a few customers who were nanotechnologists and engineers who were buying chips from us in the early days of our commercial fabrication, but we didn't really have any biologists yet. And towards the end of 2015, I was talking at the American Society of Cell Biology Conference, and we were presenting some measurements in-house that we'd been taking. And I was approached by a postdoc from Stanford, Nir Kveet, who had a problem he thought we could solve. And Nir's problem was that he had developed a peptide that was specific to delta PKC, or PKC delta. PKC delta is a kinase, an enzyme that builds up during blockage of blood flow and then creates a lot of damage once the flow is restored. It's involved in heart attacks and the damage that's created during a heart attack. Nir's peptide is meant to block the damage from happening by blocking a particular domain called the PK domain of this delta. He had proven that his peptide worked in mice, and his peptide did stop the damage in mice hearts when PKC delta was activated during a heart attack. But his goal was to show that it stopped that damage because of the targeting mechanism that he designed. And he couldn't show that because PKC delta is a very short-lived protein. This gets back to that idea of the dynamism, the dynamic nature of biology. Things change very rapidly. And most of our engineered systems, like a lot of past biosensors and the internet, rely on a stable infrastructure and stable tools. Biology doesn't work that way. This is an enzyme that's activated and stays around for a couple of minutes and then is recycled. And so when you only have a few minutes to try and measure something, the tools available to you are limited. Nir had tried to get the data in the lab showing that he could specifically target the PDK domain of this enzyme, but he failed because the tools required him to take measurements for a longer period of time than this enzyme is alive. He thought that our technology would be able to help him take this measurement much quicker because we have this ability to combine instant measurements without adding extra complexity from labels. Typically, you might use a label to do this type of measurement, but that requires development of a label and addition of more reagents and a movement away from what's really happening in the biology to an abstraction of what's happening with the label. Doing a direct measurement of whether or not his peptide bound to this kinase is what he wanted to do. So about a month after the conference, I traveled to Stanford and spent an afternoon working with Nir trying to get the data. And this is the data that we got right away. This is one of those unique situations in science that almost never happen, where you sit down to take a measurement and right away you get great data. We sat down, we attached his peptide to the surface of our chips, and we put in different solutions containing his protein PKC delta and a control protein PKC epsilon, which is very similar to PKC delta, but without the domain he designed his peptide to bind to. In this graph here on the left, the target is the PKC delta and the control is PKC epsilon. We got this data in an afternoon, and this was data that he'd been working on getting for about 6 months. So this very quickly led Nir to a bunch of other work. He finished up a bunch of projects, became our first real biologist customer, published some papers. Now he's a professor at Bar-Ilan University, and his peptides, I think, are being developed into pharmaceuticals, which is great. So this is what digital biosensors can do. They can help you gain insight into dynamic, hard-to-measure elements of biology very rapidly. I'm going to get into some more applications later, but in the very beginning, this was most useful towards researchers who had particularly hard problems. Before we get to that, I haven't yet explained how field effect biosensing works. And now that I've shown you a little bit of what field effect biosensing does, I want to show you how field effect biosensing functions. This is a diagram of our device. In the beginning, we have our same orange Pac-Man shaped proteins. That's our biology. It's the biological components, could be an antibody, could be a peptide, as in Nir's case, could be a piece of DNA. Those are chemically bound to graphene, which forms the basis for a transistor, which is just an electrical device to measure current. The sample is introduced on top of the biology and graphene, and this actually forms part of the circuit. The circuit is completed after the sample is added. Electrochemical controls are used to control the voltage in the sample, the liquid that's put on top of the biology. And that liquid could be anything. We've done whole blood, urine, or buffers like you'd find in a research lab. The biology now that's sitting in between the sample and the graphene is sitting in between a controlled voltage in the sample and a controlled voltage measuring material in the graphene. Any biochemical interactions that change how that attached protein layer, attached receptor layer is formed, anything binding or interacting with our biology attached to the graphene changes how the voltage is passed from the top, the sample part, to the bottom, the graphene part. That allows us to instantly detect biochemical interactions. We've published a few papers on this, most recently in Nature's Scientific Reports. There's some links here on this page. The Scientific Reports is open access and is a good summary of how our system works. Why does this work? I am a physicist and a nanotechnologist, and so I think that I'm required by some sort of secret physics rule to have an equation in at least every presentation that I give. So this is my required equation. Here's how this works from a physics point of view. The base of the conductor, the graphene, is our conduction channel, and that forms the basis for our circuit. What we're doing is we're changing the gate on the graphene. So in this equation above, there's a whole bunch of stuff in the beginning which relates to the really basically the resistance of the graphene, and then there's some terms inside that those parentheses that talk about what the gate is doing. The bulk solution is setting the overall gate voltage. That gate voltage is modified by two things, the double layer region and the Donin equilibrium region. In old ion-sensitive field effect transistor work, you would only have the double layer region. And that double layer region is where the different salts are moving around back and forth on top of the graphene. And it's right up close to the surface, and it's usually too small of an area to do any significant biosensing in. The Donin equilibrium region is referring to the area that encompasses all of the biology, as well as any membrane or synthetic polymer area that we use to immobilize the protein, DNA, the biological receptor to the surface. Altogether, what you can think of in this Donin equilibrium region is that there is a layer on top of the graphene that consists of some receptors and some stuff that holds those receptors in place. And those materials have different salt concentrations than the bulk solution. As salt moves back and forth due to binding of some event or some other biochemical interaction, we see a change in the Donin potential, which is a voltage that we read on the graphene as if it was an extra gate voltage. This Donin potential is the same thing that you see on a cell membrane when you don't have any sort of activity pumping it. So it's the same sort of voltage that you might get at rest in a cell. Altogether, this is what causes our gate response. And again, our most recent paper describes this in a lot more detail. This is what our biosensor architecture actually looks like. So on the left there, that's a picture of one of our biosensors. It's about the size of one of your fingers. It's pretty small, maybe the size of a glass slide. It contains in the middle a little circle. That's where you put the liquid. And on that circle, there's a micro-patterned sensor array. That sensor array contains the on-chip potential control. And as you can see here, those little blue rectangles in the middle of that picture, those are the sensors. There's 15 of them on every chip. This is where the measurements actually take place. So they're pretty small. This is our standard architecture. I'm going to show you some pictures of some of our other chips towards the end of the talk. This is what the sensor system looks like. The important thing to get from this slide really is that the size of the overall system is really about the size of a large cell phone. So it is something you can carry around in your pocket. I've carried these around in my pocket. It's meant to be a personal benchtop tool. It's very easy to use, powered via USB. There's not a lot of extra stuff you have to carry around with you. It's got some nice specifications here in terms of large dynamic range and small amount of material requirements. But the basic idea is that this is pretty small. There's more customer publications, links to them here, the people that have been publishing in addition to NIR's initial publications. I want to give you one example here of, I think, a more relevant measurement to the idea of the Internet of Biology. This is taking an ELISA and converting it to field effect biosensing. An ELISA measurement is probably the most common type of measurement done to measure a biological interaction in a diagnostic setting or a monitoring type setting. It's a labeled measurement. It means that you're going to take usually something like an antibody, you're going to bind an antigen to it from the sample, and then you're going to bind a label to that antigen. And the label might be something that fluoresces or changes color, some optical measurement that you can run. Doing an ELISA is pretty slow, but very, very reliable once it's worked out. So it's a great tool for current diagnostics. We took an ELISA for IL-6, which is inflammation biomarker. It's used in a lot of different tests. And we took that kit from Life Technologies, just a standard kit for IL-6 detection. we attached the antibody from that kit that binds IL-6 to our chips. So you can see here at the bottom, this is the typical workflow for us. You take a baseline measurement, you immobilize your receptor layer. In this case, we used an antibody against IL-6. You add a blocking layer, which fills in any gaps that the antibody may not have filled on the graphene. You run an association, meaning you try and see if you can detect the binding of the IL-6 to the surface, and a dissociation, you look for it to wash off. And those steps tell you a lot about how tightly things have bound. We used a lot of small numbers of material for this. The important thing here is we didn't use the label. So we used about half the reagents of the IL-6 kit, but we were trying to get the same result. We also wanted this to happen in 15 minutes instead of the usual several-hour measurement. This is what some of the data looks like here. This is multiple chips. This is just the immobilization. So this is putting the antibody down on the surface. And there's a lot of steps to that. You start with your baseline. You have to activate the surface of the chip. You do some rinsing. You put your antibodies down. You block it. You quench any active chemistry, meaning you basically anything on the surface that's going to react to something in the future. You react it ahead of time so that it's inert and it's not going to change anything during the measurement. And then you rinse again. It's important to note that at this step, the chip is stable. It has an antibody on it, covalently bound to the chip. And we can vacuum pack those chips and store them and then use them anytime we'd like in the future. This allows us to prep chips for any sort of interaction that we want to measure well ahead of time. In this case, we move on to the measurements. Here's what different concentrations of IL-6, the biomarker we're looking for, do when we put them onto the chip. In a normal ELISA measurement, you would be seeing individual data points, not data over time. It's pretty rare to look at data over time in the first minute of a sample being introduced to a surface. So this real-time nature of a biosensor gives us a lot of insight into what happens at the very beginning of an interaction. The important things here are that we have duplicated the limit of detection of the ELISA. So we took half the reagents, a fraction of the time, this was 5 minutes, not 15, a fraction of the time to run the FEB compared to the ELISA, and we can see the same 2 picogram per milliliter limit of detection that you get from the more worked out, more widely tested, much more expensive ELISA kit. The limit of quantitation, we can quantify a little bit broader. Really, that's because our analytical software might be a little more powerful. The real-time nature of the data gives us a little more assurance of what we're looking at, and we're able to use small volumes so we can use more material to get to a higher concentration than you can with a typical ELISA. The important thing here, again, is that we've duplicated the effectiveness of the ELISA using our system, but done so in a much shorter time period. You can also use it for some other things. This is data from one of our papers where we collaborated with the CDC on demonstrating detection of Zika. Most diagnostic tests out there look for a response from your immune system. They're trying to trigger some response in your immune system. You see, hey, is your body trying to fight this off right now? We were able to actually look and see, hey, is there Zika virus in this sample at a level, hopefully before your immune system would even start fighting it off. The sooner you can detect something, the better, particularly something like Zika. And what we were able to show is that we could detect Zika at relevant concentrations in serum using our biosensor without a label, just directly detecting it. In the paper, we also showed that we were able to distinguish between the Zika protein that we were looking at and the similar protein that's used or that's found in a common interference of Zika tests, encephalitis. Now, this is not a diagnostic. This was just a proof of concept, but it shows the direction the technology can take. We can detect viruses right away. We can detect biomarkers that are related to inflammation. This slide here shows we can detect drugs in people's in serum. So this was part of a therapeutic drug monitoring effort that was funded in part by DARPA, where we looked at taking an aptamer against a small molecule and demonstrating that we could detect it in both buffer and in serum. Detecting drugs in real time in someone's blood is really important for dosing of antibiotics, of chemotherapy drugs, and things like that. It's important often not to overdose people, but also not underdose them. And the current ways of doing these kinds of tests are really, really difficult on the healthcare system. Being able to do a real-time monitoring of the levels of drugs in someone's system would be very helpful. So this is another direction the technology is headed. That's some of the examples of the different types of technology or the applications that are already being worked on here. There's other applications that you can find out about and actually some other Lab Roots talks, and I'll get to those a little bit at the end too. So before we finish up, a little bit on how we were able to do this. If you go back to what I was saying about the nanotechnology, the different boom and bust cycles and where we were stuck 10 years ago, it was about not being able to move the devices out of the nanotechnology labs. And there's a lot in the nanotechnology literature about different ways of making devices. And what we did is we threw all that out and we started over with a fresh process, something that would work in a commercial facility. We developed something that we call our protect and expose manufacturing process. And that allowed us to get to a point where we have predictable yield, cost, and performance, the kinds of things that really you want in your biological monitoring devices. We did this. There's three main pillars. One is we took our own control of in-house graphene handling, and there's a big difference between graphene handling and graphene growth. Just having access to the material graphene isn't enough. You need to know how to use it. And because we mastered how to use it, our cost per area for graphene is less than the cost per area for silicon. So when we make devices, the amount we spend on a particular chip in silicon is higher than the amount we spend on graphene. We have moved into commercial fabrication. We actually use a commercial MEMS foundry to do our wafer construction. And we have a lot of QA. We do a lot of QA processes at multiple points in our manufacturing flow, and we automate as much of that as we can. This focus on manufacturing has allowed us to create a couple of different form factors. And the small chip size, or small transistor size, allows us to really have unlimited form factor options. The chips I showed you are the middle chip, that 3-channel chip. That's our standard one that's sold through Nanomed. We also have a multiplex 40-channel chip that we've built, and a chip that is a 16 by 3-channel chip. That one has chips that are very, very small, smaller than the fingernail on your pinky, and are designed to be used in an array. This is just an example of the different kinds of form factors that we can build chips into. This flexibility in processing allows us to start working on a lot of applications beyond the traditional laboratory use and even diagnostic applications that we've been looking at with biosensors so far. Really, the big ones here are things like field deployable sample testing, personalized monitoring devices. The rapid and complex nature of data that we're able to provide opens us up to really using machine learning in a meaningful way. Machine learning in biology is difficult because a lot of the measurement tools use labels and other abstractions away from the real biological interactions. You find out a lot about the labeling technology and not so much about the biology. Removing that and providing direct access to what the biology is doing in real time, we really think that the machine learning capabilities that are being developed right now will have powerful effect on understanding the data that comes out of our biosensors. altogether, this may lead to a future in which we are able to do in-home diagnostic testing for any number of different things. To do that, like I said, we're not building the whole internet of biology. We're building the gateway. We need partnerships to actually build out the different applications. Biochemists who understand the applications, engineers who understand devices. We have two big partnerships right now, Nanomedical Diagnostics. They've been selling the Agile R100. The story about NEAR has led to Nanomed and the use of this tool in research labs, academic labs, and pharmaceutical labs looking at early stage drug discovery. NanoSense is our other big partnership. They've developed the CRISPR chip. And this is revolutionizing the way that people think about detecting DNA, and particularly detecting DNA using CRISPR. Dr. Iran, who has led the effort there, is actually going to be talking on LabRoots. So go look for her talk after you finish this one. We have a lot of other partnerships in the pipeline, but we're not able to talk about them right now. I would be really interested in talking with anyone who thinks they have an idea and would like to partner up. And with that, I'd like to thank you for your attention and look forward to hearing from you about any questions you might have or comments. Thanks.
And thank you, Dr. Goldsmith, for that informative presentation. I'd also like to thank LabRoots for making today's educational webcast possible. As a final reminder, Dr. Goldsmith will follow up with any questions you've submitted via email. That's all for now, and thank you for joining us. We will see you next time. Bye-bye.